So, as we know that the immune system main function is to defend our body from infectious agents, pathogens which are coming from outside to our body. But apart from that, it plays a very important function in clearing out the tumor cells. Day by day, so many cancer cells they are generated inside our body, and there is an immune surveillance system that will clear out these cancerous cells. The third important function is maintaining the homeostasis between live and dead cells. We know that daily inside our body, many cells are dying, many cells are generating. Okay, so where these dead cells are going, okay, how our body removes that. So immune system takes part a very important role that it will tag them. It will tag them with the help of antibodies, and then it must be taken up by the macrophages and other dendritic cells to the site of liver and spleen. Now, what I want to emphasize is here is that if this homeostasis is disturbed, okay, like let's say for an example, the dead cells, if they remain for quite long time in our body, it will prepare antigen antibody complexes, and this will give rise to an inappropriate immune response that is called as autoimmunity. So this concept of autoimmunity was first given by Paul Ehrlich. So what is that? Whenever our immune system gets hyperactivated, it is trying to damage our own cells and tissues. So what are the reasons for that? Why our immune system is trying to kill our own cells and tissues? So the main reason is that breaking down in our self-tolerance. Somehow the self-tolerance is compromised. Okay. So what is this self-tolerance? Means our immune system actually tolerate our own antigens. Okay. It takes identity that yes, these are my antigens and I should not react against it. Okay. Or I should not kill that particular our own body cells. But when there is a breach in this particular mechanism of self-tolerance, then the damage to the cells and tissue occurs. So let's first understand that what are the different reasons for that our immune system forgets to recognize our own cells. So the first major thing is sequestered antigens. Okay, so these are actually the hidden antigens which are sometimes released inside our body because our immune system has never recognized it. So it includes myelin basic protein, it includes your corneal eye proteins. Okay, so they have been never been exposed to your immune system. Okay, so somehow accidentally if their release is there, then the T helper cell can get activated. Another major thing is that inappropriate MHC expression. I hope students are aware of major histocompatibility complex molecules. So there are three types: MHC class 1, class 2 and class 3. But MHC class 1 and class 2, they play a major role in antigen processing and presentation. Okay, so any defective expression of this MHC on our cells can lead to activation of immune cells like T helper cells. Another important aspect is that sometimes some pathogens, including bacteria, viruses, certain parasites, they share similar antigens which are found on our own cells. So that concept is known as molecular mimicry. I want to show you this slide. If you can see that there is a sequence of amino acid given here. So the blue portion actually matches with the viral protein to that of our protein present on the cells. So you see that for an example, the acetylcholine receptors of humans, it shares almost 60% of similarity with that of poliovirus viral protein 2. So what happens is that whenever we get infection of this type of pathogens, our immune system will not only try to clear out that infection, but it will also try to damage our cells which are containing this type of receptors or you can say proteins. So the ultimate result is that your T helper cells are getting activated. The T helper cells can activate cytotoxic T cells, and the cytotoxic T cells will then try to kill your own cells and tissues. T helper cell can also activate macrophages, and it can lead to the inflammation, and that can also lead to the tissue damage. T helper cell can also activate the B cell. That's how I call this T helper cell as manager cells. It controls each and every cell of your body by releasing the cytokines. So B cells then can get converted into plasma cells, and these antibodies are again towards the self antigens, which you call them as autoantigens. So this is for students that please remember that all nucleated cells of your body can have the ability to express MHC class 1 molecule. Here what you see here is that virus infected cell which are presenting MHC class 1 molecule, and it is presenting a viral peptide, the red color one. MHC class 1 molecules are always presented to the cytotoxic T cells are peptide through and that is why cytotoxic T cells, they are known as CD8. Now cytotoxic T cells will try to kill this particular cell. On the other hand, your antigen presenting cells, for example, macrophages, dendritic cells, they also possess MHC class 2 molecules through which they can present the antigens to only T helper cells. So I think this is the first event whenever any antigen comes inside your body. Your APCs are the first line of defense. They encounter the antigen, they process antigen, and then present it to the T helper cell. T helper cell upon activation can activate the other cells as well. So remember that MHC class 1 molecules are found on immune cells such as antigen presenting cells, B cells, macrophages, dendritic cells, and APCs will also have MHC class 1 molecule. This is very important to understand the structure of MHC class 1 since we are going to discuss about antigen processing and presentation. So MHC class 1 molecule is composed of two chains, alpha and beta. The alpha chain is divided into three subunits, alpha 1, alpha 2 and alpha 3. So it is the longest chain and it has a cytoplasmic tail as well which anchored into the plasma membrane. So what you see here is the surface expression of the alpha chain. Then the beta chain will join to this alpha chain which is a very smaller subunit and the genes are presented on chromosome number 15. Remember that all genes of MHC class 1 molecules they are present on chromosome number 6. But this is the exception where you will find the beta 2 gene only chromosome number 15. The MHC class 2 molecule rather forms two identical chain of alpha and beta, and alpha is again divided into two subunits, alpha 1 and alpha 2. Beta is divided into beta 1 and beta 2, and both are having the cytoplasmic tail anchored to the plasma membrane. The structure which you see here, which is prepared by alpha 1 and beta 1, that is called as peptide binding cleft. So the process that is when it attains the sufficient size, then it will bind to at this particular groove. Let's understand the basic difference between antigen and immunogen. So antigen can be any substances which can have ability to react with the antibodies or T cell receptor. Majority of antigens are actually protein in nature, but carbohydrate, lipids, and nucleic acid can also serve as an antigen. Then what is an immunogen? Immunogens are antigens only, but they have capacity to induce a specific humoral or cellular response. So basically, immunogen is an antigen and immunogen. Okay. Now coming to the antigen processing. So the enzymatic cleavage of this large antigen, because MHC molecules cannot present the large protein, antigen has to be broken down into small peptides. Enzymatic cleavage or enzymatic cleavage of protein into small peptides, then it binds to the molecule, the entire process is antigen process. It has been shown that this process is an active process and energy demanding process. So ATP is utilized in this process, and as soon as this peptide binds to the respective MHC molecule, it goes and presented in the cell surface. So that process is known as antigen presentation. So there are two types of antigens classified like endogenous antigens and exogenous antigens. Endogenous antigens include our own cell antigens, intracellular proteins that can be termed as autoantigens. This can also include antigens of viruses. Okay, so we understood that okay, viruses they are coming from outside, so they must be exogenous antigens, but it is not the case because the viruses when they infect our cells, right, they become live and then they utilize the protein synthetic machinery of your cell. So basically your cell is synthesizing the proteins, viral proteins, and then it will present it to the different T cells. So these virus antigens, they can be considered under endogenous antigens. All altered antigens related to cancer or tumor antigens, they are also considered as endogenous antigens. So they are always presented by MSC class 1 molecules to the cytotoxic T cells. So there is one figure here shown here that how the virus infection in a cell will lead to the viral protein synthesis. So we can observe here is that viral mRNA is synthesized from the viral DNA, and then utilizing the host cell machinery ribosomes, the proteins of viruses are prepared. Then these proteins will be taken up by the MSC molecules and then presented on the cell surface. Coming to the exogenous antigens, so whatever infectious agents are there whenever they come inside our body, the antigen presenting cells will engulf them and do the phagocytosis process. So all these antigens are termed as exogenous antigens. Always exogenous class 2 molecules by the APCs to the T helper cells. Since exogenous antigens they are residing in cytosol, okay, so when they will be processed in the cytoplasm, the pathway is known as cytosolic pathway. And the exogenous antigens are always processed and presented by endocytic pathway, because endosomes have the structure which take the proteins or antigens which are present in the outer environment, and then it will be taken up inside the cell. So the cytosolic pathway, and then we'll discuss the endocytic pathway. So there are four basic steps in the cytosolic pathway. First step is uptake of an antigen. So here actually uptake is not required because antigen is already present in the cytoplasm. Second is the degradation of the antigen. Then whatever antigen is there which has to be processed in the cytosolic pathway, they must undergo proteolytic cleavage in a specialized structure known as proteasomes. We will see in the detail the structure of proteasomes. Once the protein has been broken down into small peptides, then it will attach to the MSC molecule. So that step is called as antigen MSC complex formation, or you can call them as peptide MSC complex formation. Remember that only the size of the peptide 8 to 10 amino acids can attach to the MSC class 1 molecule. And step is a transporter associated with antigen processing and presentation. This molecule will help in transferring the peptide to MSC. And the last step is presentation step, or you can say display of antigen with the help of MSC molecule. So peptide MSC complex then goes to the cell surface and presents antigen to the cytotoxic T cells. Now, not every endogenous antigen can be processed by this pathway. So only the endogenous proteins which tag with ubiquitin can undergo the proteolysis. Okay, so ubiquitin is a 76 amino acid polypeptide. When it binds to the protein or endogenous antigen, then only it is allowed to enter into the proteasome subunit. So what is a proteasome? It is a big enzymatic complex. Okay, so the main unit of this complex is known as LMP, low molecular weight polypeptide. So there are different types of LMPs, LMP2, LMP7, LMP10, but LMP2 and 7, they play a very important role in presenting the antigens to or processing the antigens in this pathway. So let's discuss the LMP molecule. So it is basically a 26S protease complex which has been divided into 20S proteasome and two 19S subunits. You can see that these are the two 19S subunits at the end and at the beginning. But the middle portion of 20S proteasome actually cleaves the large antigen. Okay. So you see here how the cytosolic protein is tagged by the ubiquitin, and then it is allowed to enter into this large proteasome complex, and small peptides are generated. So as I told you that LMPs are of different types, it has been seen that LMP is actually induced by a cytokine known as interferons, and that is the reason whenever we have infection of viruses, our viral infected cells secrete cytokine known as interferon. They will activate your LMP2 and 7, and that's how the viral antigens can be processed fast. So you see that ubiquitination process is also an active process. Energy is utilized here, and then this ubiquitin is entering into this complex and peptides are formed. Now what happens is that always not the peptide of 8 to 10 are generated. Some may be a little bit larger. Okay, so at least 15% of those peptides still need some trimming. Okay, so one or two amino acids are removed to get the appropriate size of the peptide. So this function will be done by your cytosolic proteases, which are already present in the cytoplasm. So that is the one aspect. What you see in this figure is that your protein is converted into small peptides. But the another story is also simultaneously going on in the rough endoplasmic reticulum. So this is the double membrane you can see here, which is of rough endoplasmic reticulum. So MHC molecule will be synthesized in this ER. So you see that first your MHC class 1 alpha chain is synthesized, the green color one. As soon as alpha chain is synthesized, calnexin will go and attach to it because it is very unstable. Calnexin also do one more function, that it will create a binding site for the beta 2 subunit. The small subunit which you see, you can look at the cursor. So this is the beta 2M. Okay. So once beta 2M binds to this alpha subunit, the quaternary structure of MHC1 is complete. Okay. So as soon as this quaternary structure is complete, then calreticulin and tapacin, these are two essential molecules which bind to either side of this MHC molecules, and thereby it forms a bridge, right, or the support it will provide to transfer the peptides by TAP molecule. Transporter associated with antigen processing are, there are two type of TAP molecule, TAP1 and TAP2. What we see here is as a pink color. So their function is to take the peptides which are generated in the cytoplasm and bring it to the peptide binding cleft here. So these steps, they are essentially, they are only binding to 8 to 10 or maybe 8 to 15 amino acids, and then only it can be transferred to the peptides. So here you can see that calnexin, tapacin and calreticulin, all the three proteins are very important for peptide binding transfer. Now let's understand the entire pathway with this animation. You see here is a proteasome complex to which your large antigen is divided into small peptides. So basically it includes your LMP2, LMP7. So what you have now is that your peptides have been formed in the cytoplasm, and now you have MHC molecules in the ER. Okay. So this peptide must have been transferred to the ER. So TAP molecules involved here. So TAP1 and TAP2, they will take the peptides from the cytoplasm and take it to the endoplasmic reticulum. So you see that this is a synthesis of MHC class 1, alpha chain has been prepared, so calnexin will come and join to it. Then calnexin will be removed and beta 2M will join, so structure is complete. Then tapacin and calreticulin will come, and with the help of the TAP molecule, the peptide will be transferred to this particular peptide binding cleft. Sorry. Yeah. So you see that whatever the complete peptide MHC complex have been prepared here, they are ready to go to cell surface. But what about those to whom your peptides are not binding? So those MHC molecules, incomplete MHC molecules, they will be sent for degradation by the lysosome. So they will be degraded completely. We are now moving to the another pathway that is known as exogenous pathway or endocytic pathway. So this pathway is mainly for exogenous antigens. So any foreign antigens, pathogenic antigens which are engulfed by the antigen presenting cells will be processed by this pathway. So there are two types of antigen presenting cells, professional APCs and non-professional APCs. So professional APCs always present antigens. This includes macrophages, B cells, dendritic cells. But non-professional APCs, these are the APCs which are occasionally they are presenting the antigens. Okay, so it includes fibroblasts in the skin cells, glial cells in the brain, pancreatic beta cells have also been demonstrated to present the antigens. So here also you will have this four steps. So first there is an uptake of an exogenous antigen. So the cell or APC has to take the antigen inside. So for that you have three processes. It can take antigen by phagocytosis or by endocytosis or by pinocytosis. The second step is degradation of an antigen and transport of MHC class II molecule. So this will happen in a specialized compartment that is known as endolysosomes. The invariant chain plays a very important role. I will show you this thing in the figure, so there you can understand it better. So invariant chain is actually a particular protein which will bind to the MHC class II molecule, so that not a single endogenous antigen can bind to it, because we want that MHC class II only binds with exogenous antigens only. Once the degradation of the peptide and uh, synthesis of MHC class II is complete, then you will have antigen MHC complex formation. So once this complex is formed, then it is ready to present on the cell surface. What is CLIP? I will make you understand in the later slides. Okay. So first let's understand what are the differences, basic differences between phagocytosis, pinocytosis, and endocytosis. So the large solid particulate antigens, when they are engulfed and processed by APCs, this process is known as phagocytosis. It includes the complete bacterial cells, fragment of cells, etc. Pinocytosis happens when you have fluid-filled vesicles and the antigen is trapped inside those vesicles. So macropinocytosis has been seen to be done by mainly the dendritic cells. Endocytosis is uptake of any material into the cell by the membrane-bound vesicles. Okay, so endocytosis can be done for soluble as well as low levels of insoluble particle as well. So the receptor-mediated endocytosis are mainly done by many cells. So you will have a receptor through which the antigen is binding, and then the entire thing has to be taken up inside in the structure known as endosome. So you see here that there is one antigen which is trapped inside the endosome. It has come inside the cell and now it is ready to fuse with the lysosome, and peptide transfer will occur in the endolysosome complex, and then it will be presented on the cell surface. Now, in many cases of bacteria, you have many cells like macrophages and all, they will do this type of function. But B cells can also act as professional APCs because B cell has a receptor antibody. So through this receptor antibody, they will bind to the antigen, and this antigen antibody complex entirely, it will be taken up inside. Okay, so this is called as receptor mediated endocytosis. So once the antigen has been taken up inside, it will take around 1 to 3 hours to complete the endocytic pathway and to appear on the cell surface. So here the amino acid of 13 to 18 amino acid peptide, they will be only transferred to the peptide binding left. So whenever the peptides are taken up inside, that first endosome is called as early endosome because its pH is around 6 to 6.5. Then it will become late endosome because again pH will drop 5 to 6, and then it will become a lysosome. Okay, so because we know that lysosome contain highly acidic environment of 4.5 to 5. So why this gradual process of early, late and lysosome is required, so that your large antigen can be broken down into required peptide size. So I am giving an example of B cell receptor mediated endocytosis and formation of this endosome and lysosome. So you see that this is a B cell surface where you have antibody as a B cell receptor antibody. The antigen gets attached. It has been taken up through a endosome. So this endosome is called as early endosome. At the pH of 6 to 6.5, the antigen gets dissociates from the antibodies, and this empty antibody is now again recycled back to the cell surface. Now this early endosome will then convert it to the late endosome, get the pH environment of 5 to 6, and now this antigens will be degraded further in the lysosome in the environment of 4.5 to 5, right? And MHC molecules which have come into the endosome specialized compartment, they will fuse with this lysosome and the peptide will be transferred. So as I just told you that it is not just a simple process. You require a specialized molecule that is known as invariant chain. What you see here in the yellow color is the MHC class 2 molecule, and this green color structure is known as invariant chain. So what happens is that whenever MHC class 2 molecules are, they are also synthesized in the rough endoplasmic reticulum. So there may be some endogenous antigens which can partly, they can attach to it, right? So to prevent that binding of endogenous antigens to the MHC class 2 molecule, this site should be blocked. So invariant chain will block this particular peptide binding cleft. Also, it will direct, or you can say guide, this particular invariant MHC molecules to the endosome. You see that further this invariant chain is being cleaved by the action of certain endosomal proteases enzyme, and still whatever small portion you will have in the peptide binding cleft, it is known as clip. It is known as class 2 invariant chain peptide. Okay, so this clip will remain attached until unless now this MSC molecule will find the appropriate peptide. So you see that here in the endoplasmic reticulum, the MHC molecule has been attached with the your invariant chain. Then this will taken up into the endosome. In the endosome, the half portion of the invariant chain gets cleaved out, and clip will remain. Then this will fuse with your lysosomes, which are containing the processed peptides. But you can see that these red color peptides are unable to attach to the peptide binding cleft because still the clip is attached to it. So there is one another molecule known as HLA-DM. HLA-DM will come associated with this MSC class 2. It will remove the clip and allow this red color peptide to bind to the peptide binding cleft. So HLA-DM will help in the release of the clip fragment so that antigenic peptide can bind sufficiently to this peptide binding cleft. So let's see one animation also for this entire process. So first we will discuss that how the exogenous antigens are taken up inside. So what you see here is a particular antigen synthetic cell, and this is the first is phagocytosis process. So a phagosome will be prepared. If the pathogen or antigen is taken up through the fluid filled vesicles, it is called as pinocytosis. Next is receptor-mediated endocytosis. Certain APCs, they contain a receptor for Fc portion of the antibodies. We know that an antibody has two main portion, antigen binding fragment known as FAB portion and the Fc portion. So when antibody gets attached to the bacterial cell, its Fc portion is free so that it can bind to the Fc receptor on the antigen presenting cell. Similarly, APCs will have also receptor for complement proteins. So it has been seen that complement proteins can also bind to the bacteria. So this entire complex can be taken up inside, and this process is known as receptor mediated phagocytosis or receptor mediated endocytosis. And this is the example of how B cell will taken up these exogenous antigens through the receptor mediated procedure. Okay. So now we have the bacterial, APC cell surface, the bacteria has been taken up inside, then the early late endosome will be formed, and you can see that the enzymes present in those compartments will try to cleave out those large proteins into small peptides. So you see that now the peptides have been formed. Now simultaneously in the ER, you will have the synthesis of alpha and beta units of the MSC class 2 molecules, but peptide cannot bind to it because of the invariant chain has attached to it. So this invariant chain also will be cleaved out partially. So cathepsin, this is the enzyme which will degrade the invariant chain. But still you see that when it fuses with the this type of endosome and lysosome, in the endolysosome compartment the peptide cannot be transferred because this red color clip is still attached to it. So how to remove this clip? So for that we have HLA-DM molecule. So HLA-DM will come here, it will remove the clip and it will add the sufficient peptide of 13 to 18 amino acid on the MSC class 2 molecule. So what happens to the other MSC class 2 which has not bound with the peptide, or which may be empty, or which may be having still the clip attached to it, right? So those will be sent for lysosomes for degradation. But the correct appropriate peptide MSC complex will be exported to the cell surface to present the antigen to the T helper cell.